Welcome to Tech Disruptions, deep dives into the technologies reshaping industries, from AI to quantum to biotech. Let's get into it. For years, the promise of CRISPR gene editing has been this incredible molecular scalpel capable of rewriting the code of life. But it's always been hobbled by a really unglamorous problem: the actual size of the tool.
Exactly. Think of it like having the most precise micro screwdriver in the world, but it's too big to fit into the standard toolbox everyone uses. The toolbox in this case is a tiny, harmless virus that's supposed to deliver the CRISPR system into cells.
And that too big problem has meant that most CRISPR therapies to date have been ex vivo, meaning they take cells out of your body, edit them in a lab, and then put them back. It's a clunky workaround, expensive, and can't treat many diseases. But now it seems that bottleneck might finally be breaking.
Two recent breakthroughs, announced within days of each other, offer solutions to both that delivery problem and another critical challenge: how to ensure the CRISPR scissors only cut what they're supposed to, especially when dealing with something like cancer. This could fundamentally change the approach to in vivo gene therapy.
So the first problem to unpack is the delivery bottleneck. Why has the sheer physical size of CRISPR-Cas9 been such a roadblock for in vivo treatments?
It really comes down to the preferred delivery vehicle, adeno-associated viruses, or AAVs. These are nature's tiny, non-pathogenic delivery trucks. They're great because they don't cause disease, they can be engineered to target specific tissues, and they don't trigger a huge immune response. The problem is, they have a strict cargo limit, around 4.7 kilobases.
And the standard CRISPR system, the Cas9 protein, just blows past that limit?
Completely. The gene for the workhorse Cas9, Streptococcus pyogenes Cas9, or SPCas9, plus its necessary guide RNA, is too large. You simply can't stuff the entire editing system into a single AAV vector. The report describes it as CRISPR isn't the hard part in 2026, delivery is.
Which is why the ex vivo approach has dominated, right? Taking cells out, modifying them, putting them back in. It's worked for blood disorders like sickle cell, but it's clearly not a universal solution.
Not at all. That vein-to-vein model, as it's called, is incredibly complex and expensive. It's a bespoke manufacturing process for each patient. Crucially, it's useless for diseases in solid organs like the brain, heart, or lungs, or for systemic conditions where widespread editing throughout the body is needed. It's a clunky workaround to a fundamental packaging problem.
So the search has been on for a smaller, more compact CRISPR system. And this is where the first breakthrough comes in, from the University of Texas at Austin and Metagenome Therapeutics. They found something called Al3Cas12f RKK. What makes this different?
For years, researchers have been looking for naturally smaller CRISPR enzymes, often called mini-CRISPRs. They explored the vast biodiversity of microbial defense systems. This led them to the Cas12 family, which are inherently more compact than Cas9. Metagenome identified a promising candidate, called Al3Cas12f from a bacterium, which was about a third the size of SPCas9, small enough to fit inside an AAV.
Okay, but mini CRISPRs aren't new, and they've historically had a major Achilles heel, haven't they?
Absolutely. This is where the skepticism usually kicks in. The history of smaller CRISPR systems is littered with examples of enzymes that were compact but terribly inefficient. They might only work 10% of the time in mammalian cells, which is simply not good enough for a clinical therapy. The critical challenge has always been pairing small size with high activity.
So if Al3Cas12f was initially promising but still falling short on efficiency, what did the UT Austin team do to crack that nut?
This is the elegant part of the discovery. Instead of just finding a small enzyme, they really engineered it. Led by David Taylor, they used cryo-electron microscopy and machine learning to build detailed 3D models of Al3Cas12f, watching how it interacted with DNA. What they found was that this specific enzyme forms an unusually stable, tightly connected dimer, essentially two protein molecules locked together.
Like a perfectly assembled piece of machinery?
Precisely. Taylor described it as having an extra-large interface between its components, making it very secure. He said Al3Cas12f basically comes pre-assembled and ready to go shortly after its pieces are produced. This inherent stability was key. It allowed the enzyme to engage with its DNA target much more efficiently and quickly than other mini-CRISPRs.
So they understood why it was stable, and then they optimized it?
Exactly. Armed with that structural insight, they started tinkering, introducing specific mutations. They created numerous variants, and one, dubbed Al3Cas12f RKK, was the breakthrough. Its editing efficiency skyrocketed from under 10% with the original enzyme to over 80%, even hitting 90% in human cell lines. That's the critical leap, small and powerful.
80 to 90% efficiency, even for a compact enzyme, is significant. What did they target to show this wasn't just a lab curiosity?
They went for clinically relevant targets. They introduced L3Cas12f RKK into human leukemia cells and specifically targeted genes associated with complex, solid tissue diseases that are currently untreatable with ex vivo methods. This included genes linked to amyotrophic lateral sclerosis, or ALS, atherosclerosis, a major cause of heart disease, and various cancer-driving mutations.
So they proved it wasn't a one-trick pony, that it could work across diverse and challenging genetic targets.
That's right. Across all those diverse targets, the RKK variant consistently achieved over 80% editing efficiency. This really signaled that it's a robust and versatile system, not just something that works in one narrow context.
This all sounds incredibly promising. If this can be reliably packaged into an AAV, what does that actually mean for patients and the biotech industry?
It's a highly sought-after goal for many in the field. It means a potential shift from that complex, multi-week ex vivo cell removal and reinfusion process to a single injection. Imagine a vial of AAVs engineered to home in on the liver or muscle tissue, delivering this compact CRISPR system directly to the cells that need editing. It's more scalable, potentially less expensive, and applicable to a vastly wider range of diseases.
But a note of caution is warranted, right? This is still in a petri dish. What are the immediate next hurdles before this is seen in humans?
Absolutely. The researchers have proven the enzyme works with high efficiency in human cells in vitro. They have not yet proven that an AAV vector carrying that enzyme can achieve the same results in a living organism. The immediate next step, and what everyone will be watching, is the move to preclinical animal models.
So, can the AAV effectively deliver the payload to enough cells in a living animal? Will those 80 to 90% editing rates translate in vivo? And will the AAV or the enzyme trigger an immune response?
Precisely those questions. Immunogenetics, especially, is a known challenge with AAVs. The success of those animal studies will be the true test of whether this can move from a promising scientific paper to a transformative clinical reality.
Okay, so that's solving the packaging problem. But almost immediately after this news, another breakthrough drops, addressing a different but equally critical problem: ensuring the CRISPR scissors only cut the right targets, especially in something as complex as cancer. This one involved Wageningen University and the Von Andel Institute, and a system called Tumor-Hunting.
This is a fundamentally different approach. Traditional cancer therapies, like chemo or radiation, are notoriously blunt instruments. They kill cancer cells, but also cause massive collateral damage to healthy tissues. Even standard CRISPR, which targets specific DNA sequences, faces a challenge. If a cancer-driving mutation exists in a gene that also appears in healthy cells, the CRISPR tool could cut both, leading to harmful off-target effects. The core problem is distinguishing a cancer cell from a healthy cell when their underlying genetic code might be nearly identical.
So they needed a way to differentiate. How did they do that?
They looked beyond the genetic code itself and focused on the epigenome, specifically DNA methylation. Think of methylation as tiny chemical tags attached to DNA. These tags don't change the underlying genetic sequence, but they act like switches, turning genes on or off. It's a core mechanism of how the body regulates gene expression.
And cancer cells mess with these switches?
They do. Cancer cells have highly aberrant or dysfunctional DNA methylation patterns. Genes that should be active are silenced and vice versa. These unique methylation patterns become a distinct molecular fingerprint for a tumor cell, distinguishing it from a healthy cell, even if their core DNA sequence is the same. The researchers hypothesized they could use this epigenetic signature as a chemical address to guide a CRISPR enzyme.
That's a fascinating shift. Instead of reading the words on the page, they're reading the annotations. How does Tumor-Hunting actually accomplish this?
The team identified a CRISPR variant, Tumor-Hunting, and through detailed structural analysis, found it had a unique property. Every CRISPR system needs what's called a PAM sequence, a short, specific DNA sequence it must recognize and bind to before it can even begin to unwind the DNA and check for its target. It's like a docking station.
And Tumor-Cas9's docking station is special?
Exactly. They discovered that Tumor-Cas9's PAM recognition site includes a human methylation site. This means its ability to bind to the DNA is directly affected by whether or not that site is methylated.
So if the docking station is methylated in a specific way, it binds. If it's not, or if it's methylated differently, it doesn't.
You got it. The researchers use a great analogy. Think of the CRISPR enzyme as a screwdriver and the DNA's PAM site as the head of a screw. In a healthy cell, the methylation pattern might be like a flathead screw. In a cancer cell, that aberrant methylation pattern is like a Phillips head screw.
So you program your Tumor-Cas9 to be a Phillips head screwdriver,
And it scans the genome. When it encounters a healthy cell's flathead PAM site, the tool doesn't fit. It can't bind, it moves on, leaving the healthy DNA untouched. But when it encounters the Phillips head PAM site of a tumor cell, the specific methylation pattern it was designed to recognize, the tool fits perfectly. It binds securely, unwinds the DNA, confirms its target with its guide RNA, and then makes the cut.
That's incredibly precise. What did their experiments show in the lab?
They tested this theory by taking culture dishes with a mix of healthy human cells and human tumor cells. They introduced Tumor-Cas9, programmed to recognize the aberrant methylation pattern of the cancer cells. The results were a stunning proof of concept. Tumor-Cas9 successfully and selectively cleaved the DNA in the tumor cells, almost completely ignoring the healthy cells. It acted as a highly selective molecular assassin, distinguishing between healthy and disease cells based on this subtle chemical difference.
So this isn't just an incremental improvement. It's a fundamentally new way of thinking about how to target cancer, moving beyond just the genetic sequence.
That's precisely it. It's a fundamentally new approach in oncology. Aberrant methylation is a hallmark of many diseases, not just cancer, including childhood cancers and even some autoimmune disorders. This opens the door to therapies that could be exquisitely precise, hunting down malignant cells based on their unique epigenetic fingerprints, as John van der Oost from Wageningen University put it, specifically towards tumor cells.
But again, this is still in the lab. What's the reality check here? Cleaving DNA doesn't automatically mean a cure, does it?
Absolutely not. The researchers are clear about the limitations. Their study demonstrates selective DNA cleavage in a petri dish. It does not yet prove that this cleavage is sufficient to actually kill the tumor cells. The critical next phase is to show that making these specific cuts actually triggers apoptosis, or programmed cell death, in the tumors, and this happens without causing unforeseen off-target toxicity. A tool that cuts perfectly but doesn't kill the target cell isn't a viable therapy.
So if both these breakthroughs, the compact delivery system and the epigenetic targeting system, continue to prove out in animal models and beyond, what's the long-term vision? Are we talking about a combined therapy that solves both problems at once?
That's the tantalizing prospect, isn't it? The report specifically highlights this convergence of technologies as the next major leap. Imagine an in vivo therapy that combines the compact delivery of something like Al3Cas12f RKK with the epigenetic precision of Tumor-Cas9. A single tool that solves both the packaging problem and the collateral damage problem would be a monumental achievement, potentially transforming oncology and many other fields.
And what about the regulatory landscape for something so precise and systemic?
That's going to be a huge challenge. Regulatory bodies like the FDA will need to adapt. In vivo therapies present different safety and long-term monitoring challenges than ex vivo treatments. The bar for proving safety will be extraordinarily high, and establishing new guidelines for these novel modalities will be a critical step.
So these two breakthroughs, announced so close together, really do shift what's possible in gene editing. What are the key takeaways for listeners to chew on?
First, the long-standing delivery bottleneck for CRISPR, which was a very unglamorous but fundamental problem, now has a credible solution with highly efficient compact enzymes like Al3Cas12f RKK. Second, gene editing is moving beyond simply targeting specific DNA sequences. Epigenetic targeting, using systems like Tumor-Cas9, offers a new level of precision, especially for distinguishing diseased cells from healthy ones.
And that implies a potential future where in vivo gene therapy, delivered directly into the body, becomes a much more scalable and broadly applicable reality for a wider range of diseases.
Indeed, it's a roadmap to potentially curative genetic medicine that is far more precise and less damaging than current treatments. But it's also clear that there's a long road ahead, with significant validation needed in living systems.
So the biggest question now is perhaps, how quickly can these two innovative approaches converge, and what will the regulatory environment look like when they do? That's all for this episode of Tech Disruptions. Join us next time as we keep exploring the technologies reshaping our world. Thanks for listening. We'll see you next time.
Stay curious out there.